This woman is 60 years old, but her DNA believes she is 30. We aren't talking about cosmetic surgery. We are looking at a complete cellular reversal of her epigenetic profile. Is she a genetic anomaly, or have we finally cracked the code to resetting the human biological clock through protein cycling? Her name is not important right now. What is important is what her latest blood panel is telling us. Chronologically, she is 60 years old. She has lived through 6 decades of solar radiation, oxidative stress, inflammatory responses, and the relentless biochemical war that strips the human body of its cellular integrity year after year. By every external metric, she should be aging the way the textbooks say she should. But her Horvath Scale reading tells a completely different story. The Horvath clock, for those unfamiliar, is not a wellness gimmick. It is a DNA methylation-based biological age calculator developed by UCLA biostatistician Steve Horvath. It does not care how much sleep you got or whether you took your supplements. It reads the methylation patterns on your genome, the chemical tags that accumulate with age like rust on iron, and produces a biological age with a margin of error of roughly 3.6 years. It is one of the most rigorously validated aging biomarkers in clinical science. Her reading came back at 31. That is not a rounding error. That is not a favorable morning sample. That is a 29-year gap between who she is on paper and who she is at the cellular level. Her telomere length sits in the top 5th percentile for women under 35. Her inflammatory markers, specifically her C-reactive protein and interleukin-6 levels, are lower than the average 40-year-old following a clean diet. Her NAD+ levels, the metabolic currency your mitochondria burn to keep you functional, match what researchers typically see in subjects half her age. So what is actually happening inside her body? The honest answer is that we do not know for certain whether she represents a replicable blueprint or whether her genetics handed her a biological lottery ticket that the rest of us simply cannot cash. That question is the entire point of this video. But before we dismiss her as an outlier and move on, we need to look very carefully at the 3 specific interventions she has been running for the last 11 years, because the cellular mechanics behind each one are not theoretical. They are documented, peer-reviewed, and increasingly difficult to argue with. Let us start at the beginning. Let us start with protein. The fitness industry has spent 40 years telling you to eat more protein. Hit your macros. Keep your leucine threshold. Protect your muscle tissue at all costs. And for a certain kind of training goal, that advice is not wrong. But if your goal is cellular longevity rather than muscle hypertrophy, the relationship between protein intake and biological aging is far more complicated than the supplement industry wants you to believe. Here is the mechanism you need to understand: autophagy. Autophagy is your body's internal recycling system. The word comes from the Greek meaning self-eating, which sounds alarming but is in fact one of the most important survival mechanisms your cells possess. When autophagy is upregulated, your cells begin breaking down and reprocessing their own damaged components: misfolded proteins, dysfunctional mitochondria, oxidized organelles, the exact cellular debris that accumulates with age and drives the inflammatory cascades we associate with aging diseases. The Nobel Prize in Physiology or Medicine in 2016 went to Yoshinori Osumi specifically for his work on autophagy mechanisms. This is not fringe biology. This is foundational science. The problem is that autophagy requires a specific trigger, and that trigger is nutrient scarcity, particularly amino acid scarcity. The mTOR pathway, which is the body's primary anabolic growth signal, directly suppresses autophagy when it detects sufficient amino acids in circulation. Translation: every time you hit your protein target on a conventional high-protein diet, you are potentially signaling your cells to skip the recycling process entirely and just build more tissue on top of existing cellular damage. Her protocol flips this logic entirely. She runs what she calls a protein cycling window, structured across a 5-day rotation. For 3 consecutive days, she drops dietary protein to approximately 0.4 grams per kilogram of body weight, well below the conventional maintenance threshold. During this low-protein window, mTOR suppression lifts, autophagy activates, and her cells begin aggressively processing accumulated damage. On days 4 and 5, she surges protein intake to 1.6 to 1.8 grams per kilogram, specifically timed around resistance training, to capitalize on the freshly cleaned cellular environment and rebuild lean tissue without the inflammatory burden of continuous high-protein feeding. The critical engineering point here is the resistance training itself. Without mechanical stress on muscle tissue during the low-protein window, the body will begin catabolizing muscle as an amino acid source. She avoids this by keeping training volume high but intensity moderate during restriction days, preserving the anabolic signaling from mechanical load while still allowing the nutritional deficit to drive autophagy. The result, in theory and increasingly in her biomarkers, is a body that regularly flushes its own cellular garbage rather than papering over it with continuous growth signals. But autophagy alone does not explain the full picture. Cleaning the cellular interior means nothing if you do not harden the structure around it. That is where the cold comes in. Every time she steps into water at 11 degrees Celsius for 6 to 8 minutes, she is doing something that the unprepared human brain interprets as a genuine threat to survival. That is not a bug in her protocol. That is the entire point. The concept underlying her cold immersion practice is hormesis, the biological principle that low doses of a stressor, rather than causing damage, trigger a disproportionately beneficial adaptive response. The same logic applies to exercise, caloric restriction, and certain phytochemicals. The stressor is not the problem. The stressor is the signal. At 11 degrees Celsius, several things happen simultaneously inside her body. First, her sympathetic nervous system fires a norepinephrine surge that peaks at levels 2 to 3 times above her baseline. Norepinephrine is not merely a stress hormone. At the cellular level, it acts as a potent anti-inflammatory agent and has been shown in multiple studies to significantly reduce tumor necrosis factor alpha, one of the primary inflammatory cytokines associated with accelerated biological aging. Second, and this is the mechanism most longevity researchers are currently focused on, her body begins producing cold shock proteins, specifically a family of RNA-binding proteins led by RBM3. These proteins serve as molecular chaperones, stabilizing RNA and preventing the misfolding of proteins during thermal stress. The remarkable finding, first demonstrated in rodent models and now being explored in human subjects, is that RBM3 upregulation appears to protect synaptic connections in the brain and may play a role in preventing the neurodegeneration associated with conditions like Alzheimer's disease. Third, sustained cold exposure at this temperature range activates brown adipose tissue, the metabolically active fat that burns energy to generate heat rather than storing it. Brown adipose tissue activation increases mitochondrial density and improves insulin sensitivity, both of which are strongly correlated with reduced biological aging on methylation clocks. Her specific protocol is not random. 11 degrees for 6 to 8 minutes hits the precise window where brown adipose tissue activation is documented without triggering the kind of sustained hypothermic stress that begins to create cortisol spikes and immunosuppression. She does not go colder to go harder. She goes precise to go effective. 3 sessions per week. No exceptions for 6 years. But here is where the cascade gets interesting. The cold hardens the newly autophagy cleaned cells against thermal and oxidative stress. The norepinephrine reduces the systemic inflammation that would otherwise accelerate epigenetic aging. The cold shock proteins stabilize her genome at the structural level. All of that cellular work, however, still needs a physical infrastructure capable of delivering nutrients, clearing waste, and maintaining the mechanical integrity of her tissues. That is the third pillar, and it is the one that most longevity discussions completely ignore. Aging is not only a cellular phenomenon, it is a structural one. Your fascia, the dense connective tissue that encases every muscle, organ, and nerve in your body, undergoes a progressive process of fibrosis and stiffening as you age. Cross-linking of collagen fibers reduces tissue elasticity. Lymphatic drainage slows. And critically, senescent cells, cells that have stopped dividing but refuse to die, begin accumulating in the fascia and surrounding extracellular matrix, secreting a toxic inflammatory cocktail researchers call the senescence-associated secretory phenotype, or SASP. SASP is one of the primary drivers of what geroscientists call inflammaging, the chronic, low-grade systemic inflammation that silently accelerates biological aging even in people who feel perfectly healthy. Senescent cells are effectively bad neighbors. They stop contributing to tissue function, but they continuously poison the cellular environment around them. Her third intervention directly targets this structural problem. 3 times per week, she undergoes what she describes as high-intensity mechanical stimulation, specifically deep tissue percussion therapy combined with targeted myofascial release at sustained pressures between 40 and 60 pounds per square inch across major facial planes. The sessions last 40 to 50 minutes and are performed by a practitioner trained in connective tissue biomechanics rather than conventional massage therapy. The mechanical loading serves 2 functions simultaneously. First, sustained high-pressure stimulation of facial tissue creates a hydraulic flushing effect that mobilizes senescent cells and metabolic waste products from the interstitial fluid, pushing them into lymphatic channels for clearance. Animal studies using targeted senolytic interventions have shown that reducing senescent cell burden in connective tissue directly lowers circulating SASP markers within days. Her mechanical approach achieves a similar drainage effect without pharmacological senolytics. Second, cyclical mechanical loading of the extracellular matrix stimulates fibroblast activity, the cells responsible for producing collagen and maintaining the structural integrity of connective tissue. When fibroblasts are mechanically stimulated at appropriate frequencies, they upregulate collagen synthesis and reorganize existing collagen fibers along lines of functional stress, restoring elasticity that cross-linking had compromised. Her latest tissue elasticity assessments show values consistent with a woman in her late 30s. Her lymphatic turnover rate, measured via indocyanine green lymphography, sits 30% above age-matched controls following identical exercise protocols. This is the part of longevity science that the supplement industry cannot sell you in a bottle. It requires mechanical input, time, and precision. Now we have to confront the uncomfortable question that has been sitting underneath this entire analysis. Here is the skeptic's argument, and it is a legitimate one. Every exceptional longevity case faces the same critique: survivorship bias. We are not analyzing the 60 women who followed identical protocols and aged conventionally. We are analyzing the one woman whose results were extraordinary enough to warrant analysis. By selecting her as a case study, we have already filtered for success, which means her data cannot be cleanly generalized to a population. Furthermore, her baseline genetics may be doing more work than her habits. Variants in the FOXO3 gene, the so-called longevity gene, appear in centenarian populations at roughly twice the frequency of the general population. ApoE2 carriers have measurably lower Alzheimer's risk and slower epigenetic aging rates than carriers of other alleles. Telomerase activity varies significantly by genetics before any lifestyle intervention touches it. We do not have her full genomic sequence, which means we cannot rule out that she started with a 20-year biological age advantage before her protocol added another 10. But here is what the comparative data actually shows. When her biomarkers are placed against a 2026 cohort of longevity-focused individuals following broadly similar protocols, the kind tracked in ongoing citizen science databases like the Quantified Self-Longevity Project and Yale's Human Performance Lab aging panels, her numbers are outliers, but they are not impossibly distant outliers. The cohort average for biological age reduction among individuals following combined autophagy cycling, regular cold exposure, and fascia-targeted mechanical therapy sits at 8 to 12 years of epigenetic reversal. 8 to 12 years is not 30. But it is also not 0. The honest reading of the data suggests that her protocol produces a genuine, measurable, biologically plausible effect, and that her genetics likely amplified that effect rather than created it entirely. She may be an N of 1 in terms of magnitude, but the direction of the effect is replicable. So what does this mean for you? Here are your actionable takeaways sequenced by accessibility. Start with protein cycling. You do not need a lab to run a 3-day low-protein window. Drop to 0.4 grams per kilogram for 72 hours, maintain moderate resistance training volume, and then reload at 1.6 grams per kilogram on training days for the following 2 days. Run this rotation monthly and track your fasting insulin and inflammatory markers at your next physical. Build cold exposure progressively. Do not start at 11 degrees. Start at 15 to 16 degrees Celsius for 2 minutes and reduce temperature and increase duration over 6 to 8 weeks. The adaptive response requires consistency over months, not heroic single exposures. 3 sessions per week is the documented minimum for sustained brown adipose tissue activation. Address your fascia. Whether through professional deep tissue work, percussion devices, or structured myofascial release protocols, begin creating mechanical input into your connective tissue. The lymphatic system has no pump. Movement and mechanical pressure are the pump. Without it, senescent cell accumulation in the fascia is essentially unchallenged by any nutritional or cold protocol you run. And finally, get your epigenetic age tested. In 2026, consumer-grade biological age tests using methylation analysis are accessible enough that you can establish your own baseline before running any of these interventions. Without a baseline, you are flying blind. With one, you are running an experiment on yourself with measurable endpoints. She is 60 years old and her cells do not know it. The question is not whether her results are real. The question is how much of them you are willing to build for yourself. If you want the full breakdown of the epigenetic testing protocols we referenced in this video, that analysis drops next week. Subscribe so you do not miss it.